Welcome to The Explainer. Today, we're diving into an absolutely incredible microscopic journey. We're going to trace the path that starts with just 2 cells and ends with a unique genetic code that makes you, well, you.
So it all boils down to this one question, right? How on earth do we get all the complexity, all the diversity of a human being from something as simple as a sperm and an egg coming together? Well, the answer is this amazing story of migration, division, and a brilliant genetic shuffle.
The secret to all of this is written in a language that every single cell in your body knows how to read. It's your genetic blueprint, and it's all packed away on these tiny structures called chromosomes.
And here it is, the magic number for humans, 46. Pretty much all of your cells, whether it's in your skin or your brain, have 46 chromosomes inside. Think of it like a 46-volume encyclopedia that has all the instructions for building and running you. And they're always organized in 23 pairs.
Okay, so how do we get that number 46?
Well, most of your everyday body cells are what scientists call diploid. That just means they have the full set, all 46 chromosomes arranged in those 23 pairs. You get one set of 23 from your mom, the other set from your dad. But your reproductive cells, the sperm and egg, they play by different rules. They're haploid, which means they only carry a half the encyclopedia, just 23 single chromosomes all ready to pair up.
Now, to understand how we make those special haploid cells, we gotta meet the hero of our story, the primordial germ cell, or PGC for short. This little guy is the ancestor cell, the one responsible for carrying your genetic legacy into the future.
And let me tell you, this cell is a traveler. Just look at this timeline. Super early in development, when you're just a tiny ball of cells, these PGCs go on this epic migration. They actually form way back in week 2, and then for the next 3 weeks, they journey through the entire developing embryo to find their final home in what will become the ovaries or testes. It's a seriously important trip.
And as they're traveling, they're not just moving, they're multiplying. They use a process called mitosis. The easiest way to think about mitosis is as a perfect cellular photocopier. One cell splits into 2, those 2 become 4, and on and on. All of them perfect, identical clones. It's all about building up their numbers for the next big step.
You know, this migration has to be incredibly precise. These cells have a very specific destination in mind. So, what happens if some of them take a wrong turn? What if they get lost along the way?
Well, this is where things get really interesting, and maybe a little bizarre. If a PGC gets lost, it can sometimes form a type of tumor called a teratoma. And because these are what we call pluripotent cells, meaning they have the power to become any kind of cell, these tumors can contain a wild mix of tissues, like teeth, hair, even bone. It's kind of a strange but really powerful example of just how much potential these cells have. Okay, but for the cells that do complete their journey successfully, their most important mission is just getting started. They've got to prepare for fertilization by going through a really special kind of cell division called meiosis. Now, meiosis isn't just a simple copy-paste like mitosis. No, it has 2 very specific, very critical jobs. First, it has to cut that chromosome number in half from the full 46 down to just 23. And second, and this is so key for what makes us individuals, it has to shuffle the genetic deck to create totally new, unique combinations of genes.
The difference here is huge. Mitosis is just one division, and it makes 2 identical cells. It's for growth and repair. But meiosis is like a 2-act play, with Meiosis I and Meiosis II. It's way more complex, and the end result is 4 genetically unique cells, each with half the chromosomes, built for just one thing, reproduction.
So let's really focus on that second goal, because honestly, shuffling the genetic deck is where the magic really happens. It's the reason that unless you're an identical twin, there is nobody else on this planet exactly like you.
And the secret weapon for all this genetic shuffling is this beautiful, intricate process called crossover. So what exactly is crossover? Okay,
During that first big meiotic division, the paired chromosomes, the one from your mom and the one from your dad, they get super close. They literally wrap around each other and then swap pieces of their DNA. Imagine having two different cookbooks, tearing out a bunch of pages from each one and then swapping them. You end up with two completely new cookbooks with a mix of recipes. That's crossover. So, this swapping, this crossover, that's the first step in creating variety. But there's a second step. After they swap pieces, the way these chromosomes get sorted into the new cells is completely random. So the combination of crossover and then this random sorting means that every single one of the 4 final cells is totally unique. And just when you think you've got it, here comes a fascinating twist in the story. Because while the basic process of meiosis is the same for everybody, the final result is dramatically different in males versus females.
Just take a look at this. In males, it's a very efficient system. One starting cell goes through meiosis and produces 4 equal mature sperm cells. Simple, symmetrical. But in females, the strategy is completely different. The starting cell divides, but it's totally asymmetrical. It puts almost all of its resources, all its nutrients, into making one single massive egg cell. The other 3 cells that are made, called polar bodies, are tiny and just kind of fade away.
So at the end of the day, this entire elaborate process, the migration, the division, the shuffling, it all comes down to one goal. Create a gamete with 23 unique chromosomes that's ready to fuse with another. And at that moment of fertilization, the 23 from the egg and the 23 from the sperm unite, bringing that chromosome number right back up to 46 in a brand new individual.
You know, when you think about it, from that single cell on its dangerous journey to this incredible dance of chromosomes swapping genes, this process is literally the source of all our individuality. It's this amazing mix of precise cellular choreography and just beautiful creative randomness. And it kind of makes you wonder, what's more incredible? The precision that makes it all work, or the beautiful randomness that makes each of us